So I'm here to talk to you about the systematics of the Australo-Papuan Whipsnakes, Demansia. So just a little bit about me. Oh goodness, sorry, it's my first. Sorry about that. All right, just a little bit about me. I'm a PhD student at the University of Adelaide. I'm primarily studying sea snake systematics, but I'm going to talk to you today about a side project on terrestrial whipsnakes from found throughout most of Australia. So the genus Demansia are active diurnal predators of reptiles. They typically hunt through they typically hunt by chasing down their prey, which is typically skinks and geckos during the day. They are an early diverging member of the Australian Elapidae radiation. Most of their closest relatives are actually sort of rainforest dwelling species in Papua New Guinea. So it's thought that they represent an early sort of lineage into Australia that was taking advantage of the open habitats that came with drying associated with the Miocene and Pliocene. They are found pretty much throughout the whole of Australia, barring the cool southeastern corner of Australia, which is fairly inhospitable to sort of diurnal egg-laying reptiles for the most part. And there's a single species in the southern savannas of Papua New Guinea. And there's a total of 15 taxa that are now recognised, which makes them the most speciose genus of the Australian terrestrial Elapidae radiation. So historically, not many species of whipsnake were recognised. A large part of this is due to the fact that there's almost no variation in the scalation between species. So typically in snakes, numbers of ventral scales, mid-body scale rows, that sort of thing are typically used to determine species, or were in the sort of previous centuries. But in the Demansia whipsnakes, most of the obvious differences between species are actually in the colour pattern. As you can probably see here, that a lot of those colours are typically lost in preservative, particularly colours like reds can disappear, reds and greens can disappear in preservative. And the other reason for the lack of recognition of taxa historically is the fact that most species occur in the tropical northern Australia, parts of Australia, which is sparsely inhabited. And until the last few decades, there really wasn't very much collecting going on there. But in the last 15, 20 years or so, there's been a sort of explosion in number of species recognised. So it went from about 3 or 4 in the early 90s to now at 15. And there's sort of 3 main categories of whipsnake recognised. There's firstly these sort of large black whipsnakes. There's 2 species, and they're well over a metre, found throughout the sort of northern tropical region. And another group is the sort of so-called dwarf whipsnakes, which is a collection of about 7 or 8 species from, again, predominantly distributed in the monsoonal tropics, as well as in some of the sort of peripheral desert areas like the Pilbara and the Black Soil Plains. And most of these species were underneath the taxa Demansia torquata until Shane Scanlon in 2007 revised them. And other than those, there's the most well-known species groups, the Demansia psammophis group, which are sort of the well-known yellow-faced whipsnake. And the reason being is this is the group that penetrates the sort of the more populated eastern and southern parts of Australia. But despite this diversity and a bit of work being done on them a bit over a decade ago, no one has attempted to do a molecular phylogeny. There's not really any samples on GenBank, surprisingly. And in addition to that, this sort of supposedly well-known Demansia psammophis group is one that's actually quite problematic taxonomically. So you can see in this range map that there's sort of 3 main blocks of taxa recognised. There's 2 subspecies of Demansia reticulata, one in the Mesic southwestern part of Australia, one Demansia reticulata cuproceps that is found throughout the entire desert region of Australia, but the type specimen is from the Pilbara of western Australia, which is about here, and then the well-known yellow-faced whipsnake from eastern Australia. So although I've used the most commonly used kind of taxonomic scheme here with 2 species and then 2 subspecies underneath Demansia reticulata, pretty much every source has a different combination of these 3 names. Some of them recognize them all as just subspecies of Demansia psammophis. But the sort of genesis of this project, where we sort of decided to have a go at resolving it, is because in South Australia, where Demansia psammophis and Demansia reticulata cupreceps meet, they're actually sympatric in a small part of the Flinders Ranges, which sort of suggested to us that, hey, these are probably not just, allopatric species or just subspecies of a widely distributed taxon, but actually there's some significant species barriers here preventing interbreeding. So the aims of this project were firstly to resolve the phylogeny of Demansia, just to understand the biogeography and evolutionary history of this group. But also a large part was to stabilize the taxonomy of Demansia psammophis, because every year another field guide comes out with a different arrangement of this group, despite them being the most commonly seen species in the genus. And other than that, it was just to flag undetected cryptic species or evolutionary significant units within Demansia. There's been an explosion of reptile species discovered within Australia in recent decades, and a lot of those are either cryptic or they're just distributed in the sort of monsoonal tropics that have historically been poorly sampled. So to do this, we first did mitochondrial phylogenetics. So we gathered tissues from all the major museum collections in Australia, and I ran them all for ND4. We used this to guide our approach in the next step, which was nuclear sequence data, which we generated using DartSeq. And then finally, we've been using morphology guided by the results of the genetic studies to resolve this group. So firstly, I'll talk about the sort of the broad phylogeny, and basically we managed to sample about 120 individuals successfully. And one of the first things that we found when we built this tree was that most of the early divergent lineages are distributed in Northern Australia. So Demansia torquata, Demansia simplex, all these sort of earlier diverging lineages are distributed in the Australian monsoonal tropics. This is not an entirely unexpected result. Recent studies have shown that the northern tropical part of Australia has sort of exported diversity to the cooler southern part of Australia. But one of the other things that popped up immediately that was a surprise is this species here, Demansia rimicola, denoted with the stars, has two deeply divergent lineages to the tune of about 8 or 9% pairwise sequence divergence in ND4, and may not be monophyletic, although the support value is not great between those two species in Demansia flagellatio. And these two lineages are pretty well identical. They appear to be cryptic species, and they're distributed either side of a mountain range called the Selwyn Ranges on the grasslands in between. And the other sort of main point we got from this phylogeny was that we really didn't detect a whole lot of divergence between the nominal species Demansia calidira and Demansia rufescens. These two species are morphologically relatively similar, although one has a collar and the other doesn't. But you can see here there's this animal that was Demansia CF shiny, and that's one that kind of looks like it could actually be either rufescens or calidira. And we found the haplotypes were nearly identical. So that sort of flagged to us that maybe this is simply one species that varies in color pattern based on what substrate it's in, since they're allopatric. But the main sort of original goal. The original goal of this project was to resolve this Demansia psammophis complex, problematic one. And we found that Demansia psammophis, this eastern taxon, was reciprocally monophyletic. There was a bit of divergence across the Deccan Gap in Queensland between the sort of wet tropics and the more subtropical southeastern part of Queensland. But we were surprised to find that two species previously assigned to the sort of dwarf whipsnake group, Demansia angusticeps and Demansia shiny, they came out very close to Demansia reticulata, the southwestern whipsnake. We were fairly surprised by this, and the other thing was that there was this large lineage here distributed primarily in South Australia that wasn't really assignable to any nominal taxa. As the type, they were previously being called Demansia reticulata cupreceps for the most part, but the type specimen that's in the Pilbara, which means that it's probably an undescribed species. So using these mitochondrial DNA results, we've moved on with a DartSeq and the SNPs. So the SNP sequencing is primarily just focused on this Demansia psammophis reticulata group, but we are sequencing some more in the kind of broader among the other species to resolve some of the other issues that have been flagged by the mitochondrial DNA. And basically, the SNP data is largely concordant with the mitochondrial DNA. Again, we have this Demansia psammophis, Demansia shiny, Demansia reticulata as sort of good species, and then this undescribed species, which we've named Demansia spur desert. The only other thing that would surprise us was that there doesn't appear to be any support for the two subspecies of Demansia reticulata. And having gone and looked at some of the specimens now, it appears that there aren't even any morphological characters that diagnose them. It's just sort of a relic of the original description about 30 or so years ago. So one thing that is missing from this SNP phylogeny is the Demansia angusticeps. That's been sent off, but that in the mitochondrial phylogeny, Demansia angusticeps was nested within reticulata. So we're basically waiting on the SNP data to determine whether angusticeps is yet another sort of color pattern variant of Demansia reticulata, or whether it's actually a good species and it's just the mitochondria being funny, as it can do sometimes. So that sort of leaves us with this map here. We have our western shield species, Demansia reticulata, the Demansia spur desert distributed throughout the western, sort of central and western deserts of South Australia into the goldfields, and also the McDonnell Ranges of the Northern Territory. Demansia shiny, which is a sort of northern, sort of in the subtropical deserty sort of region of Tanami and southern Barkly tableland. And then finally, these sort of two lineages of Demansia psammophis across the Bedekin Gap, although we expect with better taxon sampling that may just be sort of an isolation by distance effect going on. And you'll also notice there's a major gap within New South Wales just due to delays in tissue sampling, but we've now sent samples off from that region as well. So future work, basically we're waiting on some more SNP data back. That should hopefully be back very soon, and that will resolve the sort of question with Demansia angusticeps. And additionally, morphology work's underway now. It appears that we have some pretty good characters for our undescribed Demansia spur desert. It's typically not reticulated at all. Or only reticulate a little bit, and it has a different shape to its comma. And this appears to be consistent throughout its entire range, and specimens, even museum specimens, are pretty readily assignable to that, either that species or reticulata, which is most similar. And it's also some preliminary data showing that they're typically sort of shorter, more robust, larger heads than Demansia psammophis, but I'm currently in Perth sort of filling in the other species for morphology as we speak. So I just want to thank my primary supervisor, Kate Sanders, for letting me sort of go on this side project adventure, as well as my primary collaborators and all the museum curators and collection managers that are sort of integral to doing a project like this on a widespread taxon. And thank you for listening. Cheers.